Our next speaker is Prof Randy Schekman. He will talk about tackling Parkinson's disease with basic science. The Q&A session will be moderated by Prof Lulay from the Nanyang Technological University. Prof Schekman, please.
Well, good morning. I'm pleased to be back in Singapore. I used to come every year to serve on various advisory boards, but it's been some years since I've returned. And I see new buildings and many young people, and I see the place continues to be as vibrant as ever. I'm going to talk this morning about something that is only peripherally related to the research that I've done over my career. It has to do with Parkinson's disease, with which I have become familiar, unfortunately, because of a personal connection through my wife. I'll tell you about that later. I'm going to talk first about some of the basic science about Parkinson's that we know of, primarily from some neurologic studies and genetic studies on genes that have come up in familial forms of the disease. The disease is rising alarmingly around the world. It is essentially a pandemic. The Parkinson pandemic is rising in incidence more rapidly than any other neurodegenerative disease, even faster than Alzheimer's disease. There are now almost 10 million people around the world suffering from Parkinson's, and it's estimated that it will rise to a greater number in the next decade. Parkinson's is kind of like a pandemic because it obeys no geographic boundaries. It's estimated that in the next decade, over half of the new cases of Parkinson's will arise in China. We know that about 25% of Parkinson can be explained by genetic predispositions, which I'll discuss, leaving the majority of the cases of a so-called sporadic, often late onset form that is of unknown origin. It may be environmental toxins. It may be simply better diagnostic procedures. That is still kind of the dark matter of Parkinson's disease. Parkinson's disease was first discovered or first clarified by a British clinician now 200 years ago. And for all that time, it's been considered a single disease. But it really is a spectrum disorder. Patients present with symptoms of Parkinson's disease in many different ways, at many different stages in their life. There are some that present at an early age. The actor and now activist Michael J. Fox was first diagnosed with Parkinson's disease at age 29. And 40 years later, he is still living with this disease. In his case, it has, of course, visibly affected him, but he is still alive and very, very active. He does not have dementia. About 30% of Parkinson patients develop dementia, and that often is the most debilitating aspect of the disease. In that case, Michael J. Fox can be considered a long survivor with slow development of disability and little, if any, measurable dementia. So it has an enormous range and spectrum, and yet all these people are classified as Parkinson's because of at least the neurologic diagnosis based on movement disorder. And in most cases, as I'll discuss, these individuals can be traced to a defect in dopaminergic neurons in the midbrain, a relatively small number of cells that control movement and many other emotional and executive functions. Well, the pharmaceutical industry, having attempted to treat this at one disease, has developed a number of drugs through phase 3 clinical trials, all of which have failed. There is no single treatment that reduces the eventual fate. These treatments don't slow the progress of the disease. At best, they mitigate some of the symptoms. The major treatment, which was now developed about 30, 40 years ago, is a dopamine replacement treatment, which works for several years after the onset of the movement disorder. But then as dopaminergic neurons continue to die, it loses its effectiveness. Indeed, it has secondary consequences, this medical treatment. There's another treatment, a neurosurgical procedure, where electrodes are implanted bilaterally into another region of the brain that controls the awkward movements that are often associated with progression of the disease. But that treatment only mitigates symptoms. It does not arrest the inexorable progression of the disease. And there are likely other drug trials underway now, assuming that it's a single disease, that will probably not work. We need much more understanding of the basics of the disease to understand the meaningful targets that can be approached by therapeutic intervention. Here we go. So let me now summarize a bit about what's known about the pathology of the disease. In this slide, I'll have to go back. In this slide, you see on the left sections through the midbrain of patients who've died. Under panel C, the image is of the midbrain of someone who died for other reasons. And you can see this band of a stain, which is melanin, a pigment that accumulates as a result of metabolism of dopamine in dopaminergic neurons. So there's a stripe of cells that are readily seen, even without staining, in brain sections. These cells die as one ages, but as you see on the panel, the image on the left, labeled P, in a patient who died of Parkinson's disease, the melanin-containing band of cells is largely gone. It's estimated that when a patient first presents with the movement disorder, over half of the dopaminergic neurons have already been lost. And that loss continues inexorably over the next decade or more until they're virtually all gone. About 100 years ago, a German clinician, Frederick Louis, discovered in brain sections of patients who've died of Parkinson's, as you see on the right, images of large clusters, apparently of aggregated material in dopaminergic neurons in the region of the brain in which they're clustered, called the substantia nigra. And these appear and still appear to be a hallmark of progression of the disease. They are readily seen by histologic means, but unfortunately, it has not yet been possible to visualize these structures, now called Lewy bodies, in living patients because the chemical concentration of the aggregates that promote the formation of this structure are too low to have thus far developed a successful PET scan to be able to visualize this. We can only visualize these structures, Lewy bodies, in brain sections of patients who have died. The basis of the Lewy body is believed to be a protein called alpha-synuclein, a protein that was first discovered associated with synaptic vesicles. It's a small protein, relatively unstructured, shown just in cartoon form in a stick diagram at the bottom. Mutations in an N-terminal domain of this protein have been found in familial forms of the disease that are attributable to defective alpha-synuclein. These mutations often predispose to the aggregation of this protein, and it's believed that the mutant forms cause the formation of Lewy bodies. There's another even rarer form, a genetic form of Parkinson's, where the gene is triplicated, the normal wild-type gene is triplicated, and in these patients, more abundant production of alpha-synuclein also leads to aggregation and eventually to the formation of Lewy bodies. But I would caution that the appearance of Lewy bodies is just a correlation with the disease. It is not necessarily proof of causation. This has been, I think, a problem that has plagued studies on Alzheimer's disease, the assumption that amyloid plaques really are the root cause of pathology, I think, has been a faulty assumption in Alzheimer's studies, and I am very concerned that this not be the case in studies on Parkinson's disease. It's a correlation. These aggregates may simply be a way that a cell has to sequester damaging proteins to preclude their toxicity. But in the case of alpha-synuclein, it is very clear that mutant forms of the protein really do precipitate the onset of the disease in patients with the mutations shown here. Now, some years ago, another German, actually a Swiss scientist, was able to introduce the gene for alpha-synuclein into cortical neurons cultured in the laboratory. And under conditions of culture, over the course of the next 2 weeks, he was able to observe the formation of Lewy bodies in the cell culture. You can see in an inset in the upper left-hand corner that the alpha-synuclein, labeled in this case with a fluorescently tagged antibody, highlights a very large structure, inclusion within the cell. And in similar cells examined by thin section electron microscopy, as you see in the larger image here, shown in this case in false color. Can't really see the color here, but it has false color of a slightly pinkish nature. That is simply highlighting the boundary of the Lewy body. The Lewy body includes obviously not just protein aggregates, but also organelles like mitochondria and fiber-forming proteins. It is a very complex structure. It may be initiated by the aggregation of alpha-synuclein, but it is by no means only alpha-synuclein. And so it may be too large to be degraded by the proteasome or by autophagy, and thus persists and possibly causes or exacerbates pathology. A German clinician by the name of Brock was able to classify stages in the progression of Lewy bodies in patients who died at different stages in the progression of the disease. And so the disease is now classified after the fact by the appearance of Lewy bodies at different positions in the brain. At the outset, it is believed that Lewy bodies may begin in the brain stem. Some actually have evidence that Lewy bodies may form in the gut and move progressively up the vagus nerve into the brainstem, and from the brainstem then spread further into the brain, first into regions including the substantia nigra and the midbrain, but also eventually into distal cortical regions that may affect mood and may be responsible for the progression of dementia in these patients. Again, I would caution this is all correlation and not causation, at least not yet. Now, let me talk about some rather more specific aspects of the genes, other genes that, when mutant, predispose to the development of Parkinson's disease. One of the organelles that is key to understand in the cell that is likely a part of the pathology is the mitochondrion. I think many of you are familiar with the mitochondrion, but for those of you who are not biologists, let me spend just a minute describing its structure. Mitochondrion probably originated from something similar to a gram-negative bacterium that was first swallowed up by a progenitor eukaryotic cell and found to be sustained as an organelle, looking very much like the original bacterium. The mitochondrion consists of two membranes, an outer and an inner membrane, and an interior space that's filled with ribosomes and enzymes that are involved in aspects of oxidative phosphorylation and other metabolic pathways. There are two key elements in relation to Parkinson's disease. Each mitochondrion has a nucleus, a small set of genes that have been preserved from the original bacterium that sustain oxidative phosphorylation and electron transport. electron transport, which is an essential feature of the inner membrane, is the force that drives the production of ATP. The mitochondrion is kind of the battery of the cell, and it produces the energy currency of a cell, ATP, in abundance, driven by a proton motive force that is the result of the movement of electrons and protons in opposite directions across the inner membrane. At the very end of the electron transport chain, there's a large protein complex that holds an oxygen radical as a last step in metabolism, and that oxygen radical must be preserved in intact mitochondria. Likewise, the genome must be preserved in intact mitochondria. It's so important to keep these two components in an intact mitochondrion because if the mitochondrion is damaged and if it ruptures, release of oxygen radicals can wreak havoc in the cell, can break chromosomal DNA, and even if the DNA of the mitochondrion is accidentally released into the cytoplasm of the cell, a pathway of cell death is elicited because cells don't like to have DNA in the cytoplasm. So the cells throughout our body, and indeed in all eukaryotic cells, have gone to considerable lengths to devise ways of surveying the integrity of a mitochondrion. If mitochondria are damaged, as you'll see over the next couple of slides, the cell has a way of swallowing them up and delivering them to the lysosome for degradation. So here's the pathway that is used to envelop damaged organelles, including mitochondria. It's a version of a general process called autophagy, or literally cells eating themselves. Cells have a way of gathering in damaged proteins, protein aggregates, damaged organelles into a membrane called the autophagosome, which surrounds the damage and then delivers by membrane traffic and fusion to the lysosome these particles that are then chewed up by the hydrolytic enzymes that dominate in the low pH of the lysosome. So this is an essential aspect of many cells. Indeed, many tumors depend upon autophagy for survival, and there are chemotherapeutic agents that block autophagy that are being used for treatment of certain forms of cancer. But in this case, I'm going to focus just on the crucial role of this surveillance mechanism in capturing and destroying damaged mitochondria, because there are two genes that are known in familial forms of Parkinson's called PINK1 and Parkin that are involved in tagging damaged mitochondria with little peptide called ubiquitin, and damaged mitochondria that are decorated with these ubiquitin tags are recognized, enveloped by the autophagosome, and delivered to the lysosome. So there are key experiments done by a number of investigators, principally Richard Ewell at the NIH, who have discovered the exact mechanism by which PINK1 and Parkin act to detect damaged mitochondria. These enzymes, PINK1 and Parkin, live in the cytoplasm, and they are normally widely distributed in the cell. But when a mitochondrion is damaged, and this can be done experimentally by disrupting the membrane potential across the inner membrane, Parkin inserts into the outer membrane of the mitochondrion and remains there and gathers PINK1 to its proximity, and PINK1 is a form of ubiquitin, or a ubiquitin kinase, that then is covalently linked to proteins on the surface of the mitochondrion, and that event is what causes the mitochondrion to be degraded. So these are summaries from some of Richard Ewell's work, and let me show you one of the experiments that he's published that illustrates one aspect of this decoration. So in this slide, you see in the top the distribution of the enzyme Parkin in a normal cell. Normally, Parkin inserts into the mitochondria and then is lifted out rapidly. It's only when the mitochondrion is damaged that it remains lodged in the mitochondrion. So the protein is uniformly distributed about the cytoplasm. Likewise, this little tag called ubiquitin is uniformly distributed about the cytoplasm. It's waiting to be attached to various targets. So you don't see any particular distribution of either of these molecules. However, if you take, and this is experimental, if you take a cell and you treat it with an agent that disrupts the membrane potential across the inner membrane, you can see the beginning of an accumulation of parkin about mitochondrial profiles being mobilized from the cytoplasm. And similarly, you can see the coincident localization of ubiquitin along the same profiles, in this case visualized by fluorescent antibodies, and they perfectly align. This is in the initial minutes after the addition of an agent to disrupt membrane potential. If you follow this visually over time, these puncta disappear because they're swallowed up by autophagosomes and delivered to the lysosome for destruction. So this and many other experiments have illustrated why Parkinson patients with mutations in PINK1 and Parkin are so vulnerable. It doesn't say why the vulnerability is so restricted to dopaminergic neurons, but I'll tell you why we think that's true in a moment. Well, let me tell you about another very interesting story that relates to a different gene in familial forms of Parkinson's. We now know from genomic and genetic studies that there are at least 20 different genes in familial forms of the disease. So it's genetically quite complex, and we're only just beginning to understand the fundamental function of some of these genes and why they affect the viability of dopaminergic neurons. So I'm going to tell you now about the work of a really terrific biochemist in Dundee, Scotland, Dario Alessi, and a colleague of his at Stanford, Suzanne Pfeffer, who come to some very interesting conclusions about a gene called LRRK2, LRRK2. This gene was described in the literature and genetic evaluations by Andrew Singleton some years ago. And Dario Alessi, being an expert enzymologist, particularly of protein kinases, decided to make a study of this enzyme because the sequence of the gene predicted that the gene encoded an enzyme called a protein kinase. A protein kinase is an enzyme that takes one phosphate from ATP and transfers it to another protein, and that phosphorylation event often changes the activity of the target, and I'll tell you what happens in this particular case. So Alessi characterized this enzyme, found it to be a very poor kinase with a very low activity, very low turnover number. But interestingly, the mutations in the gene found in familial forms of the disease are dominant mutations that activate an otherwise very inactive protein kinase. So a very common mutation that is known around the world is a mutation that elevates the kinase activity of the normally poor enzyme just twofold. And that twofold elevation is enough to cause such damage to dopaminergic neurons. Dario then conducted a collaboration with a number of mass spec labs around the world to discover that the kinase, the activated kinase, transfers a phosphate to another small subset of regulatory proteins that are called RAB proteins, R-A-B. RAB proteins are small molecules, small proteins, that bind GTP and use that as a trigger to change the activity of some pathway that the RAB protein interacts with. In this particular case, in studies then conducted with Suzanne Pfeffer at Stanford, they discovered that the hyperactive LRRK2 phosphorylates a particular RAB protein. And when that protein is hyperphosphorylated, it causes an arrest in the biosynthesis of a cilium on a target cell that is known in neural networks to interact with dopaminergic neurons. And that is highlighted in crude summary form on this slide. So let me tell you a little bit about what's known about the communication between dopaminergic neurons in the substantia nigra over a long distance into a higher region of the brain called the striatum, in which interneurons of cholinergic nature are connected by a pathway called the hedgehog signaling pathway. So this is a signal transduction pathway that is well known and been studied in many other contexts. And in this context, the two cells have an intimate form of communication that nourishes and sustains both cells. In the normal situation, when dopamine neurons are stressed, and you'll see in a moment why they're always under stress, that sends a signal that is recognized by receptors on this kind of antennae, a primary cilium, on the surface of cholinergic interneurons. And this is then mediated by this pathway called the hedgehog signaling pathway. And when that pathway is functional, the cholinergic interneuron responds by secreting a growth factor, glial-derived neurotrophic factor, GDNF, that nourishes and sustains dopaminergic neurons. So the two communicate. When dopaminergic neurons are stressed, they have to be calmed down somehow by receiving GDNF secreted by the cells that are communicating, cholinergic neurons. So that's normal and a known pathway. What Pfeffer and Alessi discovered is that when LRRK2 is hyperactive and a Rab protein is uncharacteristically phosphorylated, this cilium is no longer produced. And as a result, the loss of cilia interrupts this hedgehog signaling pathway and precludes the secretion of GDNF, leading eventually to the death of dopaminergic neurons. So this is a very different idea about how dopaminergic neurons may be sustained and may die in the absence of this signaling pathway. Indeed, given the genetic complexity of Parkinson's, it seems likely that there are many different insults that would eventually contribute to the demise of dopaminergic neurons, this being only one particular pathway. Now, I mentioned a moment ago that dopaminergic neurons are always under stress and are particularly vulnerable. And I want to show you an image in a moment that really encapsulates the idea of why dopaminergic neurons may be more vulnerable to mutations that otherwise would affect pathways required in all cells in our body. The normal impression that you have of a nerve cell is shown in this cartoon with a cell body and an axon and nerve terminals, perhaps several. The average nerve cell may make as many as 1,000 different connections through an axonal projection. On the other side, dendrites. So this is a typical nerve cell. But unlike the typical nerve cell, a dopaminergic neuron is enormously more complicated. Here's an image of a dopaminergic neuron taken from a neurologist, James Surmeyer, in Chicago that he provided to me. You see the cell body and dendrites, a single axon, connected to a web of terminals that make as many as 1 million different connections to other nerve cells in the striatum. So this cell is enormously complex. It extends over a long distance in the brain, and it is always active and turned on, must require a constant supply of ATP just to sustain connections between these vast number of nerve terminals in this huge elaboration that you see in this diagram. So it makes sense then that mutations that cripple the mitochondrion or that prevent mitochondrial surveillance or that fail to nourish dopaminergic neurons with GDNF or perhaps many other connections to these cells, it makes sense that this cell might be much more vulnerable to these mutations than other cells in the body that also rely on the same pathways. At least that's the current view. I am not a neuroscientist. This is all fairly new to me. Why did I become involved in and concerned with this? It has to do with a personal tragedy in my life. I was married to my wife, Nancy Walls, at a park near Stanford when I was a graduate student. Those were happy days. Here we are getting married in May of 1973. Nancy was perfectly healthy, but she noted, and we noted unusually, she had a poorly developed sense of smell, which seemed just a peculiarity, nothing to be concerned about. It turns out that many Parkinson patients eventually, when they finally recognize they have disease, recall that they, at an early age, had a poorly developed sense of smell. This is not diagnostic of the disease, but it is one of several different indications that I'll mention later on. As time went on, in our lives together, we were happy and productive, and we moved to Berkeley, where I had my career, and she was a nurse. Everything was fine until in around 1996, she presented with some unusual neurologic symptoms that were hard at first to diagnose. The most peculiar aspect of her illness was that when she signed her name, the print was tiny print. It was something that I couldn't have even done if I'd tried. It's actually a loss of fine motor control that prevented her from scribing her name in the usual manner. It's called micrographia. And when she went to the neurologist with this symptom, she said, "What could this be? It's so peculiar." And he said, "Well, it might be Parkinson's." And she said, "Well, how do I know? What is there? Is there a diagnosis?" At that time, and only until this past year, it's only been a neurologist's decision to decide whether someone actually has Parkinson's. He said, "Well, what you can do is you can take a dopamine replacement therapy and see if that diminishes the symptoms." And I'll never forget the call that I had in my office one afternoon when she said, "I just took a pill, and 2 hours later, all of the symptoms that I've had just melted away." Well, at first that was good. It looked like she had an effective treatment, but unfortunately it was then a very clear diagnostic evaluation of Parkinson's disease. The drug was effective for about 5 years, but often what happens as the drug continues, patients develop hyperkinetic movements that are often associated with the advancement of Parkinson's disease, but are also exacerbated by the medication. Later on, as her dyskinesias and other symptoms advanced, she was treated by the neurosurgical procedure that I told you about, deep brain stimulation. That worked very effectively, and it controlled many of the awkward movements that she had been suffering from, and other aches and pains all melted away. But unfortunately, it doesn't really cure the disease. The disease advanced, and within a year, it became clear that she had beginnings of a dementia, which progressed, unfortunately, quite rapidly. Nonetheless, we still had a life together, and this is a weekend at our home up the coast with my kids. As the dementia advanced, Nancy was really unable to care for herself, so I had to have daytime care. And when I traveled, I had someone with her all the time. Nonetheless, we shared moments of joy, and here is an image from the Nobel banquet where Nancy and my dad sat together smiling on this happy occasion. Fortunately, the family was with me to take care of her as she needed during that period as the disease advanced. After we left Stockholm, flying home, she went to the bathroom and came back and said, "I was talking to somebody waiting at the bathroom, and he asked what I was doing in Stockholm," and she said, "What was that? What was that thing that we were just doing in Stockholm?" So she'd forgotten. Her memory was slipping very badly. Unfortunately, as the dementia advanced, she lost her ability to communicate almost entirely. She would forget what she was saying, and she would remain silent. The disease progressed very rapidly near the end. She became at times catatonic. I was unable to give her medications. I had access, because of what I had done in my career, to neurologists around the world. She was diagnosed not only with Parkinson's, but with diffuse Lewy body disease, based simply on her symptoms, having nothing to do with the visualization of Lewy bodies in her brain. I asked what I could expect as the disease advanced. What could I prepare for, even if I couldn't treat, if she couldn't be treated, what could I prepare for? And I was always given an answer by expert neurologists, and they were always wrong. This is the frustration that caregivers have dealing with patients with such a disease. You cannot predict. There is no way to chart the path of the advancement of the disease. It's very much unlike heart disease or cancer. Really, we know so little about the disease. There's nothing that can be done. Finally, and tragically, she died in the middle of the night, likely of heart failure. I was called by her general practitioner later that day after she died, who said, well, it may be that she died because of a lack of oxygen perfusion to her heart muscle. She suffered from a hypotension, orthostatic hypotension, which often accompanies progression of the disease, and for which essentially nothing can be done. It cannot be controlled. And she died in the middle of the night. When she died, I was approached by the chief financial officer of the Sergey Brin Foundation. You'll know the name Sergey Brin. He's a co-founder of Google. And of course, as a result, is enormously wealthy, but regrettably, he has a family genetic history of Parkinson's. He has a dominant allele of LRRK2. He is a carrier of the disease, but since it's a dominant mutation, he is at some risk. His mother has the same mutation. She is also a carrier, but she is ill with Parkinson's. He is at considerable risk, and as a result, some years ago, knowing this, he began to contribute substantial sums of money to the Michael J. Fox Foundation, which is the major philanthropic arm that supports Parkinson patients in clinical research in the US. They do a little, but not very much, basic science. And Brin realized that in 200 years, nothing of great substance had been done to discover the mechanism and means of progression of the disease. And so he decided by himself that he wanted to make a more substantial investment in the basic science of Parkinson's disease, to gather together investigators interested in the molecules and the cells that mediate this terrible disease. And so out of that grew an effort that I now serve as the chair of the Scientific Advisory Board. It's called Aligning Science Across Parkinson's Disease, or ASAP. You can Google us if you wish. You'll find all the information about the program. During the first 2 years after my wife died and I became involved in this, I worked together with a wonderful PhD molecular biologist by the name of Akemini Riley. And Akemini and I together charted a path forward with the promise of substantial financial support from the Brin Family Foundation. Let me tell you about the program that we have developed that has evolved into an international collaborative research network of 163 laboratories critically organized into teams of investigators to explore what we consider to be the bases of discoveries that will eventually reveal more useful targets of therapeutic intervention. Our goal was, Kemeny and I, our goal was to develop a scientific strategy and mechanism to direct the financial donations that Brin was prepared to make to solve this terrible problem. And what we decided to do was to design a program that was brought together teams of investigators collaborating in real time rather than just individual investigators. Let me tell you about how we've gone about doing this. The problem could be considered like a puzzle. I've told you about some of the elements of this puzzle. There are, of course, a failure in protein degradation systems that may cause the accumulation of Lewy bodies. There's mitochondrial dysfunction that may lead to the loss of the integrity of this organelle in dopaminergic neurons. There is misfolding of alpha-synuclein. There are also neuroinflammatory processes that may initiate or exacerbate the lesions in the brain that lead to the demise of dopaminergic neurons. This little puzzle is obviously an oversimplification. The puzzle of Parkinson's may be hundreds of pieces that we have no idea yet how to fit together. So the challenge is to solve this problem, a problem that has bedeviled individual investigators over many decades. What about bringing investigators together in a meaningful collaboration? Can that somehow improve our prospects? So after some meetings with a number of neurologists and Parkinson's experts, we decided to focus on 4 pillars that would lead this investigation. Obviously, the first and perhaps most prominent is to further study the genetics of the disease, the 20 or more genes, the associated protein products, and their biology, their cell biology, how they normally play a role in cells and then in dopaminergic neurons. As I mentioned, there are neuroinflammatory processes that clearly are associated with progression of the disease. What are the immune systems of the body or of the brain that may mediate this inflammatory process? The dopaminergic neuron, as I've indicated, is enormously complex. Its circuitry is still not completely known. We don't know, for instance, how the vagus nerve may connect to the brain, mediating the traffic of alpha-synuclein aggregates up into the brain, and we don't know how the nerve terminals connect to those million target cells in the striatum. That's an obvious mystery that remains for basic science to discover. And finally, a mysterious set of symptoms, the prodromal symptoms, one of which is a poorly developed sense of smell, that very often precede the development of the motor disorder. One particularly diagnostic syndrome that's quite unusual is called REM sleep disorder, where patients lose their ability to discriminate sleep and, while they're sleeping, dreams from nightmares. And patients who have REM sleep disorder very often act out their nightmares quite violently. They can throw themselves out of bed. They can flail around, harming themselves, harming their partners. It's not known what causes REM sleep disorder. It's not known why it's connected to Parkinson's disease. But patients who have this syndrome present eventually with Parkinson's disease within 5 years. Almost 80% of such patients present with Parkinson's disease. We'd like to know exactly what the connection is. We'd like to have cohorts of these individuals for drug trials to see if drugs can prevent that inevitable progression from one syndrome to the disease. So Kemeny and I, after having decided on these 4 pillars, set about to make very specific decisions about the kinds of individuals and the kinds of teams that we would seek in this collaborative effort. We wanted to identify teams, including individuals who had already been engaged in a meaningful collaboration. And that's not an obvious thing. Many investigators collaborate for particular targets, particular goals. But some investigators have long-term relationships with each other where they know the give and take of a collaboration means that you can't always have your name in a prominent place on the list of authors on a paper. They know the value of collaboration, and so they reduce their ego to accommodate other individuals. These are special people. Dario Lessi is one of the best examples of that because he's made a career of collaborating on equal terms with many different investigators. So we wanted to find such teams by putting out a call for teams, and they had to demonstrate in their application how they had already been successfully integrated. We wanted to attract diverse talent from fields outside of Parkinson's, so every team had to have at least one investigator who came from outside of Parkinson. They had to have one early career person, for instance, who may not have had anything to do with Parkinson's. We We're looking for gender diversity. We have a number of women in the program. And we want these teams to have a focus on one of the 4 pillars that I illustrated in the last slide. And we also have very strong feelings about openness and sharing. The teams have to be willing to share preliminary data with each other. They have to be willing to share reagents and protocols. They have to post their papers on an archive, such as the bioRxiv or the medRxiv, as soon as they're ready to submit for publication. And we insist that every publication resulting from our funding is published in an open access format so that everyone around the world, irrespective of their interests, has access to the fruits of our labor. So a snapshot of where we stand now, almost 5 years after that first initial effort. We have 35 teams, 163 investigators. We have committed now over $500 million to these teams just for the first 3 years of their investigations. 53 early career investigators actually have lead roles in the teams. 14 countries are represented. 80 different institutions around the world are involved. Each team has to have PIs who are at least at 2 different institutions. So it's quite diverse, and yet we think that diversity builds strength. Well, we've only had a few in-person meetings. We started just before the pandemic began. Our first in-person meeting where all the PIs that we had recruited appear together here in a picture in the Bahamas, quite a nice meeting. Many of these people, though working on similar problems, had never met each other. So this was a fantastic meeting. It was the first meeting that many people had gone to since the emergence from the pandemic. So it was a thrilling meeting. Here we are in the front row. This is my partner in crime, Akemini Riley. And we have representatives of the Michael J. Fox Foundation with whom we work hand in hand to organize this massive effort. This is a billion-dollar experiment. We are grateful to Mr. Brin for his generosity, for his insight. He is the unusual billionaire. He's completely hands-off. He leaves everything to us. So we feel blessed to have this opportunity. Out of this, we believe new discoveries will be made that offer insights and targets for therapy. However, for the time being, we are not funding drug discovery. We are not funding clinical trials. We are focused entirely on basic science because of my and many of us in the audience share a conviction that is through basic science that the real discoveries of impact will be made in the future. Thank you.
That was a very informative talk and raising awareness of the importance of Parkinson's disease. Right. Parkinson's disease is also very relevant to a country like Singapore because we are a country that is getting older. So according to statistics, in Singapore, every 3 persons out of 1,000 people over 50 years are having Parkinson's disease. So it's really, really relevant to our country. Actually, for the majority of developing countries, relevant. Just remind the audience that Professor Randy Schekman's Nobel Prize winning work was focused on very basic research. Actually, you are using the yeast, budding yeast, that is used in for baking the bread to study the membrane trafficking pathway. And Professor Randy Schekman's lab has identified almost all the basic core molecular cellular machineries involved in protein trafficking. So just as a very straightforward question. So what do you think that for the basic research like membrane trafficking can help in our understanding of disease like Parkinson's disease?
Well, the connections are there even before I started this effort. It's known, for instance, that mutation in one of the Rab genes, the first of which we discovered actually in yeast over 40 years ago, is in a subset of familial of patients who have the disease. The lysosome is a critical target of interest and traffic to and from the lysosome, which involves the secretory pathway. Also, appears to be essential for the survival of dopaminergic neurons. The development of damaged mitochondria by autophagosome membranes also involves membrane traffic. So, in fact, perhaps by coincidence, membrane traffic plays an intimate role in not only in Parkinson's, but in the many studies that we're funding through our program. So that part of the neuroscience I know about. The rest, I'm still on a steep learning curve.
Yeah, sure. So as Parkinson's disease is just one of a few or several neurodegenerative diseases, so there are other diseases like Alzheimer's disease. So do you think that other diseases like Alzheimer's diseases should be studied using the similar approach as you proposed?
Yeah. Well, it all depends on having a benefactor like Sergey Brin to create such a program. I will say that I've watched from a distance the Alzheimer's field, and I feel that progress has been held back by the dominance of this idea that it's the amyloid plaque that is the basis of the disease. That view has been so dominant that it has unfortunately suppressed alternative views of the origin and progression of Alzheimer's. And the drug treatment that's now available and has just been approved in the US by the company Biogen is an immunotherapy that removes plaques from the brain, but the evidence of its efficacy is very poor. And I think it was, frankly, not a good decision to approve that drug because it will be available to anybody on Medicare who has Alzheimer's disease, and it could bankrupt Medicare. The evidence from those studies shows that these antibodies do remove the plaques, but for the most part, that doesn't change progression of dementia in these patients. So I think the conclusion that people would like to draw about plaques being the primary source of pathology in Alzheimer's is simply wrong. So I hope to avoid that roadblock in Parkinson's by not assuming that the Lewy body is the thing that needs to be treated. It's something of interest, but it isn't going to be the complete focus of our study. And I think if there were something in Alzheimer's like this, real progress could be made, but it requires somebody unique like Sergey Brin to start such an effort.
Okay, now let's, due to time constraint, let's just take one question from the audience. Yes, please.
Hi, thank you so much for a fantastic talk. My name's Elise Facer-Childs. I'm from Monash University in Australia. Really interesting about the link with REM sleep disorder and generally, sleep disturbances and circadian misalignment. Do you know of any research looking at treating sleep disorders or circadian misalignment in the early stages of the disease and where that could go?
I am not aware of that, and it would be really interesting if such studies showed a diminished progression to Parkinson's, if the disease really could be treated. But I've never heard of anything like that. One thing that I can say doesn't really address your question, but bears on this REM sleep disorder. One of the organizations that we fund through ASAP is called PPMI, Parkinson Progression Monitoring Index. And over several years, this organization with clinics around the world has collected thousands of Parkinson patients, collected genetic, genomic, neurologic, blood samples, saliva samples, cerebrospinal. So there's a huge library of material that's being investigated. And just in the past year, what has grown out of this effort, which is really quite exciting, is the observation that in cerebrospinal fluid of patients, even with just REM sleep disorder, not yet progressing to Parkinson's, one finds something that causes the aggregation of alpha-synuclein. And so now there's a commercial kit to evaluate that. And the correlation is very high. And so the hope is that the PPMI, with its clinical outreach, can collect a large cohort of RBD, REM sleep disorder patients before they progress. And these would be ideal targets for therapeutic intervention with drugs that are being used in clinical trials to see if there's any diminished progression from REM sleep disorder to Parkinson's. So this is really this is a big breakthrough because, as I said at the outset, Parkinson's until this year has relied on neurologic evaluation for its diagnosis. There has not, unlike cancer and heart disease, there has not been a biological marker until the discovery that I just told you about. So the fact that patients with REM sleep disorder have this is really quite remarkable, even if they don't have any Parkinson's symptoms. So that's really hopeful.
Thank you.
Okay, due to the time constraints, I encourage the audience to ask Randy during the break time. Now I have to close this session. Now, let's thank Randy for his wonderful talk.